Hello, I'm Takahiko Shiba from TMDU.
Hi, my name is Takeaki Sudo from TMDU.
Today, we would like to introduce our recent research. It is known that dental calculus can cause dental caries and periodontal disease. In periodontal treatment, removing the dental calculus is most important aspect. However, it has been reported in recent years that DNA from dental calculus can reveal the bacterial composition that existed in oral cavities in previous eras. Japan is an island country, and the Edo period from 1603 to 1868 was a time of national seclusion called sakoku.
In this study, we investigated whether there is a difference in the bacterial composition between the people of the Edo and modern periods, as well as the prevalence of periodontal disease and the detection of causative bacteria of periodontal disease during the Edo period. We first collected 12 skeletons from Edo period and conducted clinical examinations such as alveolar bone resorption and dental calculus deposition. In order to perform microbiome analysis, dental calculus was carefully collected from skeletons, and DNA was extracted and amplified. We then decoded the sequences with next-generation sequencer and performed taxonomic analysis and network analysis using a supercomputer. In addition, we developed a new diagnostic approach for periodontitis in skeletons using micro-CT. Specifically, we first created a virtual alveolar bone crest, and the bone resorption rate was then measured by the position of the virtual alveolar bone crest, the apex of the root, and the bottom of the bone defect. We used this formula to determine the bone resorption rate. Skeletons with an average bone resorption rate exceeding 10% or bone loss of 5 mm or more was defined as periodontitis, and 5 out of 12 individuals were diagnosed as having periodontitis by this new approach.
First, I would like to show you the percentage bacterial composition at the phylum level in samples during the Edo period. Firmicutes was the most dominant phylum in the Edo samples with and without periodontitis, followed by actinobacteria and proteobacteria. There were no major differences in the phyla between the Edo samples with and without periodontitis. Next, I would like to show you the bacterial compositions in samples during the modern period. The most abundant phylum in the modern samples with periodontitis was Bacteroidetes, followed by Fusobacteria and Firmicutes. The most abundant phylum in the modern samples without periodontitis was Firmicutes, followed by Bacteroidetes and Actinobacteria. As you can see, the bacterial compositions of the Edo and modern samples were dissimilar. More detailed analysis was done at the species level in only periodontitis group. The number of species common between the Edo and modern periods, Edo-specific and modern-specific species were 17, 7, and 192 respectively. Focusing on bacteria that was detected in the Edo sample with periodontitis, the bacteria that caused modern periodontal disease were not detected. Among specific bacteria in the Edo sample with periodontitis was Egasterolenta, which can cause polymicrobial intra-abdominal and anaerobic bloodstream infections. Bacterial network analyses were conducted in order to clarify the co-occurrence relationships among each species. Porphyromonas gingivalis, that is known for causative bacteria of periodontitis, was a core species in the bacterial network of the modern sample with periodontitis. On the other hand, Eubacterium molecules, Treponema sokransky, Egasterolenta, might be important in the bacterial network of the Edo sample. The structure of bacterial networks and the kind of bacteria that constitute the network differed between the Edo and modern samples with periodontitis, suggesting that the causative bacteria species of periodontitis and the relationships between bacterial species differed in the Edo and modern samples with periodontitis.
the dental calculus can preserve bacterial DNA and reveal the microbial composition in ancient samples. The modern bacterial composition of oral cavity might be different from that of the Edo-era, suggesting that periodontal pathogenic bacteria and its co-occurrence relationships might have changed with time and environment, including the Sakoku period. That meant severely limited relations and trade between Japan and other countries. Thus, our study sheds new light on the evolution of oral microbiome and on periodontal pathogenesis.
Thank you for watching our video.